Dear students, after studying this particular module, you shall be able to learn and know about the meaning of chromatography. You shall be able to know how different chromatographic techniques are characterized. You shall be able to understand different modes of chromatography and also learn the applications of gel filtration chromatography. So let us introduce this topic of chromatography to you first. The word chromatography comes from the Greek word chroma, which stands for color, and graphing, to write. The technique was first used around the beginning of 20th century to separate plant pigments with easily visible colors. It has long since been possible to separate colorless compounds as long as there are methods for detecting the same. So you can understand, students, how chromatography has formed the base for as an important technique in the chemical world or the chemistry. Partition chromatography is based on the fact that different compounds can distribute themselves to varying extents between different phases or separable portions of matter. One phase is the stationary phase and the other phase is the mobile phase. The mobile phase means that it is the moving phase. The mobile phase flows over the stationary material that is chosen by the chemist or by the performer of the experiment and carries the sample to be separated along with it from the mobile phase. In chromatography, the sample is applied at one end of the porous support which holds the stationary phase, which may be solid or a liquid, and the mobile phase, which is a liquid or a gas. The various constituents in a sample get separated between the stationary phase and the mobile phase. The partition or distribution coefficient, denoted as Kd, is defined as the ratio of concentration of a compound in two phases at equilibrium. For two such immiscible phases A and B, the value for this coefficient is a constant at a given temperature and is given by the expression Kd is equal to the concentration in phase A divided by the concentration in phase B. Different compounds vary in their distribution coefficient in a given biphasic system, and this constitutes the underlying principle for their separation. Movement of those substances in sample which have greater affinity for stationary phase is retarded, since they cannot move along with the mobile phase because of their less affinity towards the mobile phase, and they tend to spend more time in this phase, that is the stationary phase. On the other hand, students, the compounds with relatively higher affinity for the mobile phase, they tend to move along with the mobile phase at a much faster rate, as these are carried alone with flowing mobile phase. So, after having described to you chromatography as well as partition chromatography and various terms which are used in chromatography on a daily basis, let us classify chromatographic techniques. The various chromatographic techniques have been classified depending upon the forces or the interacting phenomenon between the solute molecules as well as the stationary phase. Let us now discuss about the modes of chromatography. Chromatographic separation may be achieved by two basic techniques. First is the column chromatography, in which the stationary phase attached to a suitable matrix, an inert insoluble support that is packed into a glass or a metal column, and mobile phase passed through the column either by gravity feed or by use of pumping system or applied gas pressure. This is the most commonly used mode of chromatography by chemist people or the chemists. Now you can see here how the column chromatography is performed, and this is beautifully depicted in this particular figure that we have shown you here. Second mode of chromatography is the thin layer or planar chromatography, in which The stationary phase attached to suitable matrix is coated onto a glass, plastic or a metal foil plate. The mobile liquid phase passes across the thin layer plate held either horizontally or vertically by capillary action. Planar chromatography includes paper chromatography, in which stationary liquid phase is supported by cellulose fibers of paper sheet. Now students, let us discuss about size exclusion chromatography after having discussed the modes of chromatography. Size exclusion chromatography, also called gel filtration chromatography, separates molecules, as it is obvious from the name, on the basis of their size. It is a form of column chromatography in which stationary phase consists of cross-linked gel particles. The gel structure contains pores of a varying diameter up to maximum size. The test molecules are washed through a column of the gel, and molecules larger than the largest pore in the gel are excluded from the gel structure. Smaller molecules, however, penetrate the gel to a varying extent depending upon their size, and this retards their progress through the column, and this becomes a way to separate them. Elution, therefore, is in the order of decreasing size. So, what are the separation process being involved of components of a sample during gel filtration? Let us now discuss this aspect of gel filtration chromatography. In a sample containing a mixture of large and small molecular weight substances is applied onto a column. Solvent, usually a buffer, is used as an eluent. As components of the sample travel down the column, the compounds whose molecular size exceeds the fractionation range of the gel are unable to enter into the gel particles and hence remain completely excluded from the beads and travel through the interstitial spaces. Molecules which possess smaller compounds or molecules of smaller compounds, however, they diffuse into the gel matrix through the pores and get distributed between the mobile phase inside as well as outside the gel particles. These thus follow a longer path than the larger molecules, and hence their movement down the column is basically retarded. Consequently, different components in the sample get separated from each other, with larger molecules getting eluted first, followed by smaller molecules. For a given type of gel, students, the distribution coefficient, as I described to you earlier, denoted by Kd, which represents the fraction of the liquid within the gel particles accessible to molecules of a given substance, will predominantly depend on the molecular size of the compound. Very large molecules, due to their exclusion, have no access to the mobile phase within the gel, hence have a Kd value of 0. Whereas smaller molecules for which the inner mobile phase is completely accessible have a Kd value of 1 or unity. Here you can see a figure in which we have depicted the separation of solutes during the gel filtration chromatography due to molecular sieving process. And you can see the gel matrix and the large and small molecules and how they get separated. For the molecules of the intermediate size, that means the molecules which are not very large and not very small either, the Kd values ranges between 0 to 1. The difference in Kd value of various compounds in the sample accounts for their separation during gel filtration. The relationship of Kd with elution volume Ve is given as Ve is equal to V naught plus Kd Vi. Here students, Ve is the elution volume and represents volume of the mobile phase required to elute the compound from the column. V naught, students, is the void volume or the volume of the mobile phase outside the gel particles. Vi is the volume of the mobile phase which is present inside the gel particles and can be calculated from the relationship Vi is equal to A multiplied by Wr, where A is the dry weight of the gel and Wr is the water regain. Kd is the fraction of Vi accessible to the particular compound, and we get Kd is equal to Ve minus V0 divided by Vi. As it can be noted, for very large molecules which are completely excluded from the gel, Ve is equal to V0, and so Kd is obviously equal to 0. But for smaller molecules which can freely diffuse into and out of the beads of the gel, Ve is equal to V0 plus Vi and Kd is equal to 1. Now students, let us discuss the topic of gel filtration media. The characteristics which are desired of the gel filtration media, they include number 1, chemical inertness of gel matrix. That means that the gel media should not interact with any of the compounds which are being separated. Second is the low content of ionic group. Third is the uniform pore of the particle size. And fourth is the wide choice of gel particle and pore size. Fifth and the last one is high mechanical rigidity. There are a variety of products available for gel permeation chromatography, and they are usually classified according to their exclusion limit. That is, the relative molecular mass above which all the molecules are excluded from the gel structure. 5 types of gel fulfill these criteria to varying degrees. The gel particles are usually in the bead form, and earliest gel developed were consisted of the carbohydrate polymer like dextran or polydextran, referred to by trade name as Cefadex. Dextran is a linear polymer of glucose. The polyglucose units are basically cross-linked by epichlorohydrin into various classes of gel beads with exclusion limit of 1 and 2 lakh Daltons pore size, is controlled by molecular weight of dextran and amount of epichlorohydrin used in the preparation. These gels are identified by a number of G10 or G200, which refers to water regain value of the gel multiplied by a factor of 10. These gels are strongly hydrophilic and smell considerably in water. And before a column is prepared, a gel must be fully hydrated with a large volume of liquid in it. Second is the polyacrylamide gel. The second type of gel medium is produced by polymerizing acrylamide into a bead form. The cross-linked structure of these polymers produces pores in the material. And here you can see we have shown the structure of the same, of the polyacrylamide. The extent of cross-linking can be controlled to select a desired pore size. They are also hydrophilic and swell significantly in aqueous solutions, but chemically more stable than the dextran gels, which we have discussed a little bit earlier. These gels are sold under the name of Biorad-P and are usually identified by a number such as P10 or P100, which indicates the exclusion limit of the gel in thousands of Daltons. Large pored polydextran and polyacrylamide gels can be used to separate solutes only up to 3 lakh Daltons and may be difficult to use owing to their lack of mechanical rigidity. As a result of being soft, they tend to compress in the column, causing unacceptably slow flow rates. These problems prompted development of agarose and agarose polyacrylamide gels. Now let us discuss a little bit about agarose gels. Agarose is a polymer of glucose joined by 3,6 anhydro linkage. You can see the structure of agarose gel here we have drawn in this figure. Agarose gels are useful when gels with a very large pore size are required. They are also hydrophilic but usually sold in the swollen form. Beads of agarose are not covalently polymerized but held together by hydrogen bonds. In spite of this, these gels are quite resistant to hydrogen bond breaking agents such as urea and guanidine hydrochloride, raising the possibility that additional unidentified forces are responsible for maintenance of their structural integrity. Problem with agarose gel, students, is that they soften at a temperature above 30 degrees Celsius. So that is a very narrow range of temperature to work with. Now, the next which we'll be talking about is a mixed gel which containing polyacrylamide as well as agarose. Mixed gels of polyacrylamide and agarose are available, in which the polyacrylamide provides a three-dimensional structure which supports the interstitial agarose gels. Glass beads. Now let us discuss about these. The last medium for gel filtration chromatography to be discussed is the controlled pore glass beads. These fine glass spheres are manufactured to contain large numbers of pores within a narrow size distribution frame. Their total rigidity permits high flow rates without seriously compromising resolution. In addition to pore size, particle size also influences the resolution attainable. The smaller the bead size, the greater the resolution that can be expected. Increased resolution results from an increased surface area of gel being made available to the molecules moving down the column. This provides many more opportunities for a molecule to diffuse into the matrix with ensuring retardation. However, the increase in resolution is acquired at decreased flow rates provided by smaller particles. For most analytical applications, 100 to 200 mesh beads are sufficient. Mesh refers to the particle size of the particles which are available. Now students, let us put our focus onto the elution parameters. The effluent volume is the most convenient parameter to measure in gel permeation chromatography, because flow rates are often variable, making the use of retention times unsuitable. The sequence of different solutes emerging from column will therefore be reported as the total volume of solvent that has emerged from the column when the substance appears in the effluent, that is VE, as discussed earlier too. Molecules larger than the exclusion limit of the gel will be eluted when a volume of solvent equal to the void volume, that is V0, leaves the column, and similarly those molecules can gain access to all parts of the gel matrix will have an effluent volume equal to the total bed volume, which is V1. Molecules of an intermediate molecular size will be eluted in volumes between Vt and V0. Here we have shown you a graph showing variation in absorbance and effluent volume. Pay attention students, here the graph is basically plotted between the effluent volume and the absorbance and how it changes. The measurement of effluent volume is not very reliable because of the effect of the geometry and packing characteristics of any column. It is often useful to use a reduced parameter such as VE upon V0, which is not so dependent on the column characteristics and is comparable with the calculation of RF values in thin layer chromatography. It is possible to calculate a partition constant, that is KD, or the partition coefficient, for a solute in terms of the access gained to the gel matrix. This is possible by the following formula, which is VE is equal to V0 plus KD multiplied by Vi. Here Vi is the gel inner volume. Hence, KD becomes VE minus V0 divided by Vi, as we have discussed earlier in this module also. However, because it is difficult to measure Vi precisely, the equation must be modified to determine the available part of the resin, that is KAV. Thus, KAV, which is the available part of the resin, can be calculated by the formula VE minus V0 divided by V1 minus V0. In order to separate two solutes satisfactorily, their values of KAV must be significantly different from each other. Now students, let us discuss sample application in chromatography. Three properties that must be considered with regard are size, viscosity and ionic strength in this particular case when we are discussing sample application. First, we will discuss about the size factor. Sample size depends greatly on sample characteristic and kind of separation desired. For desalting purpose, the sample size may be as large as 10 to 25% of the bed volume, but for separation of macromolecules, a sample size of 1 to 5% is advisable. Second factor is the viscosity factor. Increasing the viscosity of the sample above that of eluents results in broadened and skewed elution profile. As you can see here, we have shown you in this figure variation of absorbance with respect to change in effluent volume and NaCl concentration. In practice, the viscosity of the sample and eluent should never differ by more than twofold. Ionic strength. Ionic strength of the sample as indicated above for eluent should be greater than 0.08 to overcome any ionic groups that may be present in gel medium. In most cases, sodium or potassium chloride is used at a concentration of 0.2 to 10 molar to adjust both the sample and eluent to appropriate ionic strength. Molecular sieve chromatography depends largely on a mixture of molecules being given an opportunity to diffuse into a particular gel matrix. The greater the number of opportunities for this to occur, the finer the resolution achieved, therefore the flow rate of the eluent exerts a profound effect on the resolution characteristics of a column. Now finally we will discuss the application of gel filtration chromatography. Gel filtration phenomenon has been used for several purposes which are briefly described to you here in this module. The first application, students, is the desalting of group separation. In a number of investigations, as during preparation of nucleic acids and protein purification, inorganic salts or other low molecular weight reagents are extensively used, which often have to be removed from the final preparative compound. This can effectively be achieved by passing preparation through the column of Cephadix G10 or G25. Second is the concentration of dilute solution. Dry Cephadix, that is G25 coarse, when added to a dilute solution of high molecular weight substance, absorbs water during the swelling process. The high molecular weight substance, however, remains in solution due to their complete exclusion. Third is the fractionation or purification. The availability of a large variety of gel media, for example dextran, polyacrylamide, agarose, and also note we have given you a table which states many other gels which are used in the market and available in the market too, with varying fractionation range, has extended the use of this technique for separation as well as for purification of biological materials ranging from low molecular weight compounds like sugars, polymers like proteins and nucleic acid, to superstructures such as viruses. Fourth is the molecular weight determination. As we have mentioned to you earlier also, separation of various components in the sample is based on their differences of the molecular size that they possess. Larger the molecular weight of the compound, lesser will be the elution volume. In fact, this method has been exploited for determination of molecular weight of the solute, particularly proteins, from their elution characteristics, that is VE or KD value. This method of molecular weight determination is simple, inexpensive, and does not require homogeneously purified preparation of the protein or compound of interest, and it is also non-destructive in nature. Now here you can see a plot or a graph showing a relation between molecular weight and elution volume during the gel filtration chromatographic technique, and you can see it for the proteins being separated. Molecular sieve chromatography depends largely on mixture of volume molecules being given an opportunity to diffuse into the gel matrix. I would like to reiterate the fact that students, molecular sieve chromatography depends largely on the mixture of molecules being given an opportunity to diffuse into the gel matrix. Again, I would like to state that the greater the number of opportunities for this to occur, the finer is the resolution that is achieved in basically separating the compound into a more purer state by this chromatographic technique. Now, after having studied gel filtration chromatography in a lot of detail and studying all the factors which affect it and various gels which are available in the market these days, and how chemistry has progressed in achieving so many gel based and different in structure and different for each type of use which a chemist desires, I would like to summarize this module. In this module, we have taught you that chromatography is based on the fact that different compounds can distribute themselves to varying extents between different phases and separable portions of matter. One phase is the stationary phase and the other phase is the mobile phase. The mobile phase flows over the stationary material and carries the sample to separate along with it. Different compounds vary in their distribution coefficient in a given biphasic system, and this constitutes the underlying principle for their separation. Size exclusion chromatography, also called gel filtration chromatography, separates molecules on the basis of their size. It is a form of column chromatography in which stationary phase consists of cross-linked gel particles. The gel structure contains pores of varying diameter up to the maximum size. The characteristic desired for gel filtration media include the chemical inertness of the gel matrix, low content of ionic groups, uniform pore of particle size, wide choice of gel particles and pore size, and high mechanical rigidity. Agarose is a polymer of glucose joined by 3,6-anhydro linkage, and these gels are useful when gels with a very large pore size are required. They are also hydrophilic but usually sold in swollen form. Mixed gels of polyacrylamide and agarose are also available, in which the polyacrylamide provides a three-dimensional structure which supports the interstitial agarose gels. So I hope, students, that this part of chromatography module you have found very interesting, as it holds a very promising technique in chemistry to separate and filter out molecules of interest in high grade of purity.